Hello guys, I'm Sneha S from Sri Venkateshwara College of Engineering. Today we'll be talking about a topic from biology that is enzymes. So what do you think an enzyme is? Let me first give you a basic introduction about a cell. The entire human body is made up of cells. There might be 3 trillion or million billions of cells in our body. So in every cell, there is a chemical reaction that is taking place. So now let us leave this biology and whenever, in chemistry, whenever we want to fasten any chemical reaction, we use catalyst, right? What is a catalyst? A catalyst is nothing but a chemical that fastens or speeds up the process of the reaction. That is, in chemistry, it's particular a chemical reaction. But in a human body, there are biochemical reactions taking place. So these enzymes are biological polymers that catalyzes a biochemical reaction in a human body. That means whenever a biochemical reaction is taking place in a human body, these enzymes are those polymers that speeden up the biochemical reactions in our body. The majority of enzymes are proteins with catalytic capabilities crucial to perform different processes. Metabolic processes and other chemical reactions in a cell are carried out by a set of enzymes that are necessary to sustain life. So, these enzymes are basically proteins and they have the capability that they can fasten any chemical reaction or they can help you perform any chemical reaction. And these are very important to sustain life. These enzymes are found in all the tissues and fluids of our body. The catalysis of all the reactions taking place in a metabolic pathway is carried out by the intracellular enzymes. The enzymes in the plasma membrane governs the catalysis in the cells as response to the cellular signals. You know, the neuron and all the entire body of, human body is made up of cells, and these cells even have this neurons. Mm, so whenever you have, for example, think two cells have to communicate with each other. So how does the other cell knows that the cell wants to communicate? As they don't have any language, there are cellular signals through which these enzymes, communicate or talk to each other. And the next one is the enzymes in the circulatory system. They regulate the clotting of blood. Most of the critical life processes are established on the function of enzymes. Now let us see the classification of enzymes. The first one is oxidoreductases. The next one is transferases. Next is the hydrolases. Then is the lyases. Then is isomerases and ligases. These are the 6 enzymes. We'll be studying each of them each of them briefly. The first one is oxidoreductases. So the name itself tells that there is any oxidation or reduction process that is taking place here. So this catalyzes oxidation and reduction processes. Oxidation is adding of oxygen, leaving of carbon dioxide, and so on. It can be giving electrons, removing electrons. So example is pyruvate dehydrogenase, catalyzing the oxidation of pyruvate to acetyl coenzyme A. And then the next one is transferases. That means there's some transfer. So here the catalyzed transfers a chemical group from one compound to another compound. One example for that is transaminase, which transfers an amino group from one molecule to another. Let us see the third one, that is hydrolysis. This catalyzes the, hydrogen bond. That means it breaks a hydrogen bond. Hydrolysis of a bond in the sense breakage of hydrogen. For example, the enzyme pepsin hydrolyzes peptide bonds in proteins. The next one is lyases. This catalyzes the breakage of bonds without catalysis. That is aldolase. It's an enzyme in glycolysis which catalyzes splitting of fructose 1, 6-biphosphate to glyceraldehyde 3-phosphate and dry hydroacetone phosphate. The next one is isomerases. They catalyze the formation of an isomer of a compound. Example, phosphoglucomatase catalyzes the conversion of gluco-1-phosphate to glucose-6-phosphate, where the phosphate group is transferred from one position in the to other position in the same compound. Next is the glycogenolysis, where glycogen is converted to glucose for energy transfer. Next is ligases. Ligases catalyze the association of two molecules. For example, this DNA are made up of 2 strands. So whenever there are 2 strands, they should have a bond through which they are connected. So they have this bond of phosphodiester bond, and this is initiated with the enzyme called ligases, sorry. Mm, the last one, cofactors. These cofactors are basically not enzymes. They're non-proteinous substances that associate with the enzymes. A cofactor is essential for the functioning of an enzyme. Enzyme with a cofactor is called a holoenzyme. These cofactors do not, they are non-proteinous, but they enhance any biochemical reaction. So they are basically associated with enzymes, so they can work efficiently together. The last one. That's all, guys. Thank you for watching. Please like and subscribe to my channel, and please hit the bell icon for more updates. Thank you.